In this video, we're going to focus more specifically on the steps of mitosis. And remember that we can use the mnemonic of PP-MAT to help us remember the order of those steps of mitosis. So PP-MAT stands for prophase, prometaphase, metaphase, anaphase, and telophase. We will also talk about cytokinesis, which starts happening at the same time that telophase is kind of finishing up. And remember that mitosis, what we're talking about during these phases, are really the chromosomes are going to be separated. So we talked about how during the cell cycle, so during the S phase of the cell cycle, remember that was our DNA replication phase. And we remember that we talked about how our bar-shaped chromosomes started to look like X-shaped chromosomes because we made a copy of the DNA. Well, during mitosis, during the prophase, prometaphase, metaphase, anaphase, and telophase, what happens is that those two chromatids actually separate from each other. So one chromatid goes to one side of the cell, the other chromatid goes to the other side of the cell, and eventually those chromatids become the new chromosomes of the new daughter cells that are produced. We'll start by talking about the first phase of mitosis, which is prophase. And there are a couple of important events that happen during prophase. First of all, something called the mitotic spindle starts to form. And the mitotic spindle is made from these organelles or structures called centrosomes. And a centrosome is made up of two of these kind of cylindrical structures called centrioles. And those centrioles have these protein fibers called microtubules that kind of sprout from them. So if we look at this picture over here, we can see that this is a centrosome and this is a centrosome. And inside the centrosomes, we have the centrioles, which are these little structures here. And then the microtubules are these little lines that are coming off of the centrioles. Now, the purpose of this mitotic spindle is that these microtubules are ultimately going to reach out and connect to the chromosomes. And so these kind of string-like proteins are what are going to pull the chromosomes around and then ultimately pull the two sister chromatids away from each other so that each new cell that's created during mitosis gets one of those sister chromatids. As that mitotic spindle is forming, the chromatin also starts to coil tightly. All right, so the sister chromatids start to become visible. So let's take a look at this picture down here. So this is actually looking through a microscope, and this is a white fish blastula, which is basically cells that are in embryonic development in a fish. And so what we're looking at on this picture here, so this line here is the cell membrane of a cell. So you can see that we've got multiple cells that we are seeing in this picture here. Now, in the cells that are not dividing, so if we look at this cell over here, this cell is actually a cell in interphase. And I can tell that because remember that interphase is this phase kind of before mitosis has occurred. And a couple of things to point out here is that it has a fairly clearly defined nucleus with a nuclear membrane surrounding it. And inside that nucleus, we can see that chromatin. And that chromatin is fairly disorganized, so it has not formed those bar-shaped chromosomes yet. Now, in the picture that's actually labeled as prophase, you can maybe compare and contrast the difference between those two cells. So in this cell, where we have our prophase going on, you can see that the nuclear membrane is not super clear anymore. And that's because it's actually started to break down. And that's something I didn't have on the list here. So we'll add that and say that the nuclear membrane starts breaking down. Because remember, the chromosomes are, or the chromatin, the DNA, it's stuck in the nucleus. So at some point, in order to separate those chromosomes, we're going to have to remove that nucleus, remove that barrier, so that those chromosomes can get separated. And that starts to happen during prophase. Now, you can also see that the chromatin has started to condense. And so what we're seeing here are things that are, maybe not quite chromosomes yet, but they're starting to look like chromosomes. So the chromatin is condensing and starting to form those chromosomes. Up here in the picture, you can also see that. You can see that the nuclear membrane is breaking down. And the chromosomes are starting to become visible, forming that X shape. Our second stage of mitosis is prometaphase. And so during prometaphase, that nuclear envelope disappears completely. So you can see on this picture over here that you no longer see the nuclear envelope. So there's no longer a nucleus or a membrane surrounding the nucleus. The chromosomes become more visible. It's a little bit hard to see because they're all kind of smushed together kind of towards the middle of the cell, but all of this kind of dark stuff here, these are the chromosomes. And you can also pretty clearly see the spindle fibers. So these are those microtubules, which are just these like stringy proteins. So these spindle fibers are just made out of the specific protein known as a microtubule. And that's what these little lines are here. And then the centrioles would be found on either end where those microtubules are connected to. You can see that on the image down here as well. So you can see the centrioles. You can see those microtubules making up the mitotic spindle. And you can see those X-shaped chromosomes. Now, each of these chromosomes has a special binding region called a kinetochore. And the kinetochore is what those mitotic spindle fibers actually bind to. And that's how the mitotic spindle is able to actually kind of tug these chromosomes around, and then ultimately is going to separate or kind of pull apart those two sister chromatids. The third phase of mitosis is known as metaphase. And what happens with metaphase is that those chromosomes line up right along the kind of the middle of the cell. So these kind of dark structures here are the chromosomes. And then you can see those kind of microtubule spindle fibers coming out from those centrioles attaching to those chromosomes. Notice also that that nuclear membrane is still gone. Now we still have a cell membrane, okay? So this barrier out here is the cell membrane, but there is no nuclear membrane because we got rid of that we started getting rid of it during prophase, and then it went away completely during prometaphase. The fourth phase of mitosis is anaphase. And what happens during anaphase is that those X-shaped chromosomes, remember our chromosomes have those two sister chromatids, those X-shaped chromosomes get split in half. And one of the sister chromatids goes in one direction, and the other sister chromatid goes in the other direction. So our X-shaped chromosomes now are getting converted back to looking at like those bar-shaped chromosomes. So you can see in the picture here, so this picture is kind of like early anaphase. And then this picture is looking at kind of like later anaphase. Where you can see these dark kind of structures here. These are those chromatids, all right? So the X shape is no longer visible. The chromatids have now separated from each other. And you can see that down here on this picture as well. You can still see those kind of spindle fibers coming up from the centrioles as well. And it's those fibers that are actually pulling the chromosomes away from each other. Notice that the nuclear membrane is still gone and the cell membrane is still there. The cell also starts to elongate, so it starts to go from being kind of like a round cell to being a longer cell. And this is important because in the next step, the cell is actually going to start to divide in two. The fifth phase of mitosis is known as telophase. And what happens during telophase is that those chromosomes are now reaching opposite ends of the original cell, and then that cell is going to start to break into two. So you can see these two pictures down here. You can pretty clearly see the chromosomes. And you can see that they're reaching opposite ends of the cell. So the picture on the bottom is kind of like an early telophase, maybe a late anaphase, early telophase. And then the one up top is a little bit later. So this is a late telophase. Towards the end of telophase, those chromosomes are going to start to decondense. So the chromosomes are going to go back to becoming the form known as chromatin. So they're going to be less visible and less organized. The mitotic spindle is also going to start to break down because it's done its job. It's separated the chromatids into two new nuclei, so we don't need it anymore. And then a new nuclear envelope is going to form around each set of the chromosomes. So we're going to start to see, and we don't really see it on the pictures here, but ultimately a new nuclear membrane is starting to form around these new sets of chromosomes that have been separated from each other. This picture is also showing you the beginning of cytokinesis. And so cytokinesis starts happening towards the end of telophase. And what happens during cytokinesis is that the cell membrane starts to pinch in. We can see that really well on this top cell here. So you can see that the cell membrane is starting to pinch in on either side. And ultimately what's going to happen is that it's going to pinch this original cell into two in order to form two new cells. Cytokinesis is the last stage of mitosis. And as I mentioned, it starts at the end of telophase. So telophase and cytokinesis kind of start happening simultaneously. And so remember that mitosis, so the PPMAT steps of mitosis, so the prophase, prometaphase, metaphase, anaphase, and telophase are specifically where the chromosomes are separating. And two new nuclei are formed. During cytokinesis, everything else gets separated. So those two nuclei are now going to be divided into two new cells. The cytoplasm is going to get divided between the two new cells, and the organelles are going to be divided. So we didn't really talk about it very much, but during interphase, in addition to the DNA being replicated, the cell is also making additional copies of the different organelles. So it's replicating the ribosomes and mitochondria and endoplasmic reticulum because each new cell needs those organelles in addition to a new nucleus and new genetic information. So the two new cells that are produced are called daughter cells. Now, this does not mean that the cells are female, okay? So these are not male or female cells, other than the fact that if they're in a female, then they're obviously female cells, and if they're in a male, they're male cells. But they don't have anything to do with sex or reproduction or anything like that. So they're called daughter cells, but really they should just be called like new cells. So daughter does not mean that they're female. It just means that it's one cell became two cells. So to form these two cells, what happens is you have this contractile ring that forms, and you have the cell membrane that starts to pinch in. And this is a really cool picture over here, in my opinion, because you can see that kind of pinching in of the cell membrane really clearly. And what happens is that the ring of proteins continues to constrict until ultimately it cuts this one cell into two separate cells. And this kind of pinching in of the cell membrane is called the cleavage furrow. So the cleavage furrow is this pinched in area that ultimately ends up cutting the original cell into the two daughter cells. I like to use this diagram to kind of summarize what happens during mitosis. And in reality, this diagram also kind of shows you what's happening during interphase as well. So this first circle represents a cell in interphase. And remember, in interphase in humans, the cell has 46 total chromosomes, which we can also say is 23 pairs of chromosomes. We could also say that the cell has 46 total chromatids. Because remember, at this point, the chromosomes are linear. So each chromosome is just made up of one bar of information. So we are going to draw just two chromosomes, or two pairs of chromosomes, I should say here. So in our picture here, we're actually looking at 4 total chromosomes, or just looking at 2 pairs. And we've got 4 total chromatids because each chromosome is made up of one chromatid. So 1 chromosome equals 1 chromatid. Now this first arrow here represents the S phase of interphase. And remember that in the S phase, we have DNA replication occurring. And so what happens is that those bar-shaped chromosomes are now going to look like an X because each one of them had a complete copy made. So after the S phase of interphase, the cell still has 46 total chromosomes. And those chromosomes are still arranged in the form of 23 pairs. However, we have now doubled the number of chromatids present. So 46 times 2 gives us 92 chromatids. And that's because one chromosome is now equal to 2 chromatids. And remember that the chromatids are identical to each other. So these chromatids are identical because they are exact copies of each other. Now the second set of arrows refers to mitosis. And so this includes the PP mat stages of mitosis plus cytokinesis. And so this is where those chromosomes are condensing, the nuclear membrane is breaking down, the chromosomes are moving to the middle of the cell, and then the sister chromatids separate. So that's kind of the key event that's happening during mitosis, is that the sister chromatids are separating. So what we end up with are daughter cells that look identical to the original cell. All right, so remember that these sister chromatids separated. And so one went into one daughter cell, the other went into the other daughter cell. Same thing with the blue chromosome. One went into one daughter cell and one went into the other. Same thing with the second set of chromosomes, where each chromatid goes into one of the daughter cells. So ultimately what we end up with, so our end result is that we end up with two daughter cells. And these daughter cells are genetically identical to each other and genetically identical to the original cell. So each daughter cell is back to having 46 total chromosomes, which are arranged as 23 pairs of chromosomes. There are 46 total chromatids, and that's because each chromosome is now made up of only one chromatid.